Hi, so today we're going to be learning about transcription and translation. You've kind of learned a little bit about transcription already, but we're just going to fine-tune that and move on to translation. We are making proteins. So we're going to learn how DNA is going to be transcribed into RNA and how RNA is going to code to make your have your amino acids together to make your polypeptides, proteins. Okay, so this should look really familiar. We have DNA and RNA. We're going to start off with DNA. So we'll have deoxyribose nucleic acid. It has a deoxyribose as the sugar. It's double-stranded, which means all your A's are bonded with T. All your C's and G's are bonded. And then you have thymine. We're going to turn DNA into RNA, right? It's going to be read and then transcribed into RNA. Ribose is your sugar. It's going to be single-stranded, and all your T's are going to be replaced with U's. Types of RNA. So we have our messenger RNA, mRNA. It's created from a DNA template and carries the information from the nucleus to the ribosomes in the cytoplasm. So this is the one, and yes, I know this is upside down. I have no idea, but you get the gist of it. So this is your DNA strand, right? So there's the types of RNA. Essentially, they're going to help with transcription translation. They're not all just RNA. So the first one is going to be the one that's produced in the ribosome. So this is the one that you guys made yesterday in your foldable. So the mRNA, this is going to happen inside the nucleus. The RNA polymerase will attach to the DNA and make your mRNA. So that's the mRNA, the messenger RNA, is what directly comes from your DNA. The ribosomal RNA or rRNA interacts with cells, ribosomes, to make them functional, so they're kind of all embedded within the ribosome, and they're just there as little helpers. And then we have transfer RNA, tRNA. That's what's actually going to come in. So this was like the messenger RNA. It's going to come in, that's the yellow part, carrying each amino acid due to a code, attach, and then leave behind your amino acids. Okay, so that is your tRNA. Okay, so we're going to start with transcription. So transcription is where our DNA comes to RNA. It's the same language, but almost like using a different pen color. So I don't know if that sounds okay. It's very similar. It's just a little different. So DNA to RNA. All right, so transcription. Segments of DNA serve as a template to produce complementary RNA molecules. This is what you guys did yesterday in your foldable. This is what that diagram looks like, not flipped. So this is your RNA polymerase. Your DNA is going to come in and make your messenger RNA. So now we know not just RNA, it makes your mRNA. All right, so step one, RNA polymerase, which is your enzyme, binds to DNA, and it will separate each strand. So each strand is going to separate from each other. So all your A's and T's will separate. And that's why we made it like a flap open yesterday. So just imagine that flap opening. Step two, it's going to use a DNA as a template to assemble the nucleotides into a complementary strand of RNA called mRNA. All thymine will be replaced with uracil. So here's your DNA strand, and this is your complementary strand of mRNA. You guys did this yesterday. Okay, and then now we're already ready to go to translation. That's pretty much all that happens in transcription. So this is called the central dogma of molecular biology. DNA going to RNA, RNA making your protein, right? This is what's happening in transcription, and this is translation. When DNA, it's nucleotide, will be basically transcribed to a different type of nucleotide, RNA, and then your nucleotide will then be translated into an amino acid. So this kind of just is a visual to show you what's occurring. So translation. So the ribosome so now the mRNA has left the nucleus and is now going to attach to your ribosomes. So ribosomes use sequence of codons in mRNA to assemble amino acids into a polypeptide chain and make proteins. The sequence of 3 nucleotide is a codon for a specific amino acid, and this all occurs in your cytoplasm. So this would be like your ribosome. This is your mRNA. It's going to come out here and attach. And then now you're going to have tRNA is going to come in with your amino acids. Every 3 nucleotides, so AGU, is a codon for a specific amino acid. All right, so step 1, the ribosome attaches to your mRNA molecule in the cytoplasm. So this right here, the mRNA, is going to come in and attach to your ribosome, which is this green structure. Step 2, as each codon passes through the ribosome, so every 3, so AGU, the tRNA will bring a proper amino acid to the ribosome. So like once like AGU passes, your tRNA is going to come in. This is all this yellow part is the tRNA, and attach, bringing a specific amino acid with it. It's like bringing it in. So the starting codon is always AUG. So when you're looking at the codon, AUG is going to start the process. And every single tRNA has an anti-codon. So the UAC, like that's like this part. It's almost like a puzzle. So like it's like, oh, okay, it's like we fit together because they're complementary. So each tRNA has 3 unpaired bases that are complementary to the codon on the mRNA. So AUG, that means like this one would have UAC. It's going to come in with this particular amino acid. Here's some examples. Like there are 64 possible codons, but there's an example of 6 amino acids. I put this like on your notes, and you're going to try to help you to identify. So this is like the codon. So you're pretending like AUG, GCA, GCA, CGA, and CCU. So like that would be your codon. So what would be your amino acid? So you're just going to use this chart to help you. Here's another codon chart. This one's a little crazier. You would use the corner first, and it would help you like work your way out, basically. We will go into this one tomorrow, and we'll go into this one. This is a different type of codon chart to figure out what kind of amino acid that you would use. And again, we'll go into this more. That's what we'll do tomorrow, is we'll spend a whole day on it. Okay, so a summary of protein synthesis. First thing is a portion of DNA unwinds and mRNA molecule is transcribed. mRNA leaves the nucleus and enters the cytoplasm where it binds to a ribosome. The previously transcribed tRNA molecules bind to specific amino acids to line up in order of their codon and how they appear on the mRNA. And the ribosomes form polypeptide bonds between the amino acids and releases the completed protein when a stop codon appears. And I'll talk to you guys tomorrow about what the stop codons are. Okay? So that's it. 8 minutes for transcription translation. That's pretty fast. Tomorrow we will practice translation, probably the next 2 days. We'll do some activities to help you guys to really get comfortable with it. I know it's kind of a lot, but hopefully you understand the idea of the process, that you're going from DNA to RNA and from RNA to making proteins. Okay, thank you.